really layer upon layer of regulatory control on top of itself. The other lesson with cancer is you don't sort of get this boom of massive aneuploidy or number of mutational changes at once, but rather cancer is actually found to sort of accumulate mutations over time, and really it's not until the acquisition of a driver mutation where things really start to take off, where you start to get a highly unstable genome, highly unstable cell, and The other compounding factor, not only are these tumors hit or the tumor cells hit in multiple ways, they then spin off progeny, and these progeny then go on to evolve in their own way. It actually results in tumor masses like this, where you have a blue cancer cell that has then perhaps given rise to a red subclone and a yellow subclone. You can actually see these microscopically, maybe not so great here with the light, but really what's happening here is you now have cells that inherit the cancer genome alterations from the parental cells, but then go on to acquire their own resistance mutations or their own genetic drivers. So you can do a therapy that's very effective against the blue clone. There's potential that the red and yellow clone may still persist. There's a need to monitor and overcome the drivers that are active in those subclones as well. Actually, a nice sort of diagram of this published 6 years ago now is really this idea of starting with your tumor clone, the acquisition of multiple clones over time. So each of these clones is defined by different cancer genome alterations. Might have a yellow mutation here, might have a number alteration here, might have a rearranging here in purple. The idea that then you have chemotherapy acting on all of these clones at once, and the risk that a resistant clone may be somewhere in that larger cellular population that may then persist and then have a space in which it can then grow and acquire additional cancer genome. So the real challenge here to not just look at cancer genomes at one point in time, but really having the ability to sample multiple samples of the actual tumor tissue, or also later a little bit of work in cell-free sequencing, really trying to read these subclonal populations and resistance mechanisms out more routinely, rather than trying to rely on diagnostic material for 4 or 5 years prior. The other challenge is these tumors will also change and evolve as they move throughout the body, as they metastasize. So in this case, some work by Peter Campbell in the UK, really starting with a primary tumor and then seeing metastatic masses that then, just like in clonal evolution, start to acquire additional cancer genome alterations. Because this primary tumor spins out of secondary metastasis, spins out of tertiary metastasis. And this is really where clinical interpretation is extraordinarily challenging. First of all, how do we sample all of these metastases? So we may maybe want to get there. But also, what do we do about have to treat this then? Or is there some clonal truncal mutation really dead that can blow away all of this? And certainly in the BRAF mutation example I showed you, that was very effective. You do see these dramatic responses, but then you also see these dramatic resistances as well. Why do next generation sequencing, or why do molecular testing for cancer in general? Obviously, treatment, we want to see what are the genome alterations, what type of therapies may work. Are you already resistant to a drug? Are there specific drugs that you cannot metabolize? Is there resistance to metabolism? I'll talk a little bit later on about an inherited hereditary cancer syndrome. It's really where you're born already with a loss of function mutation of the tumor suppressor, really with a very, very high predisposition to cancer, very early onset cancers in these patients specifically. And prognosis, what type of cancer do you have? Even if there's not a goal to assign patients to a targeted therapy, what we've seen with increasing number of tumors being sequenced and analyzed is that there are multiple really exactly like an oil pipeline. You want to have your text file at the beginning. You want to run it through a series of software modules, and sort of a pipe module. But essentially, you want to run all these software programs in a systematic way, end to end, to go from raw DNA sequencing, a raw DNA sequence to a list of variants at the end. Building one of these also very much like an oil pipeline. You want to really run through a series of software that are all compatible with each other. You want the software to really link up end to end without leaks. And you want to put them together in a really systematic way. And I'll have a slide basically each of the steps. Really the idea of taking the first sort of that text file, align it to something, some kind of reference. In this case, we use a human genome reference sequence. Anyone can download this off the internet. Of course, I'm copying my laptop, and you probably will by the end of this workshop as well. Pre-processing, really sort of cleaning up that alignment in areas that are very difficult to align. I'll show you an example of that. QC metrics. Did all your reads align to human? Did you do a human experiment? Do you have a bunch of other sequences that are not necessarily human? Did the experiment work? Does it look like the cancer you are studying? Variant calling out mutations, translocations, cognitive alterations. And I'm just putting slightly basically in gray at the bottom. How do we interpret these variants, and how do we write a clinical report to communicate this analysis to clinicians who may act on it? Really, the go-to place for almost every pipeline, certainly I've worked on, is some modification of the genome analysis toolkit best practices. And this is sort of basically a more technical version of the slide I just showed you. Really, going from raw reads, going through an alignment, going through some cleanup of that alignment, some variant calling, and then basically tidying up those variant calls. So this is actually described in great detail on the Broken Institute's website. And another great tool to bring all your attention to is the Picard suite of tools, which is really a collection of QC tools that really look at the output of this pipeline and allow you to compare it across drugs. This best practices workflow actually came out of a lot of the non-cancer work being done really across the world. And for cancer genomes, we've inevitably had to modify this and bring in our own cancer-specific colors. So very challenging to take tools that were written specifically for germline analysis and apply them specifically to cancer. I've just listed some of the tools here for doing variant calling specifically for cancer, and then also the types of cancer genome variation that we're looking at in cancer that may not be certainly as frequent in germline or normal cell genomes. So this is really step one. Basically, if you took that big text file I showed earlier, aligned all those reads to the human genome reference, this is really what you get. This is a software called Integrative Genomic Viewer. I think there's a little tutorial session specifically on how to use this tool. I'll show you a lot of screenshots of how this works, or how to read these screenshots. But just to orient you, the chromosome is here along the top. So just like the chromosomes I showed you earlier, they've taken that little banded karyogram and put it horizontally along the top. So we're looking at this portion of this chromosome. In this case, we're only looking at 92 base pairs at a time. In white is the human genome reference sequence, and then gray is a histogram of the number of reads aligned to each base. These are all the sequencing reads. This is the data I showed you on that slide earlier, and you can already see that data represented in this way is a lot more human readable than it was just in the flat text format. First of all these reads have more or less the same sequence. I've intentionally chosen the region that doesn't have any variation. So in this case, all these regions basically match perfectly to the human genome reference. And now we want to run callers that look for differences between our data and reference. And there's actually a Wikipedia article called Short Read Sequence Alignment. There are many, many ways to do alignments. BWA is the one I've used here, but there are many, many others. So really you want to choose a specific line or appropriate to your biological question. This is a cartoon of the exact type of data I just showed you. So the human genome reference being here along the top. Here's chromosome 1, chromosome 5. Here all the sequencing reads. Here are the types of genome variation we want to find using bioinformatics. Point mutations, single mutation difference from the reference, an indel, insertion or deletion of a base or multiple bases in a small region. Missing DNA, so tumor suppressors in regions that don't have any reads whatsoever. If you don't put DNA into your sequencing device, you're not going to get reads out. That region is not represented. That looks like a deletion. A hemizygous deletion, so one of the two chromosomes is deleted. That means you get half as much data as you'd expect. A copy number gain, some of these alpha genes can get amplified to tens or hundreds pieces of DNA sequencing reads for that reason. And translocation breakpoint, where you take one piece of one gene, attach it to another piece of another gene. And this now has brought one gene on chromosome 5 under regulation, under the control of regulatory machinery that are meant for gene on chromosome 1. So these are very powerful oncogenic drivers in cancer. I'm not going to talk too much about pathogenic sequences, but really not everything aligns with the human genome sequence, especially in cancers like cervical cancer, for example, that where they're one of the original drivers of those cancers is exposure to virus, HPV, EBV, etc. In this case, those reads, of course, come through your DNA sequencing reads, and you can align those to other other references on the human genome. And here's sort of a cheat sheet of sort of the ways that we can think about and sequence DNA, RNA, start to infer the presence of proteins that DNA is wrapped around. Whole genome sequencing, this is put all the DNA into the sequencing device and sequence it. It's whole genome sequencing. Exome, this is focusing specifically on exons, so only the coding portions of these genes. Much cheaper. It's probably 1%. Looking at 1% of the genome relative to the whole genome. Targeted gene sequencing, this is what we often will use in clinical labs. Much smaller targeted panels, much more cost-effective, much easier to interpret. Targeted variant genotyping, this is getting even more focused, looking at single bases, single mutation. Very high throughput, very inexpensive, the least comprehensive test, but certainly the most clinically actionable. And epigenome modifications, really looking at methylated promoters or other regulatory regions. RNA sequencing, looking at the sort of the functional output of DNA, looking at transcriptome sequencing, either looking at whole RNA sequencing. Targeted RNA sequencing is also a protocol that's being increasingly used in clinical labs, and microRNA sequencing, looking specifically at small microRNAs that are essentially confer regulatory control on gene expression. And I'm not really going to talk at all about protein mapping or DNA fingerprint mapping. Actually, may be an entire bioinformatics workshop on this topic. I want to show you real data as often as possible. So here's the exact same sample sequence using genome sequencing, exome sequencing, and RNA sequencing, focused on a very important cancer gene, KRAS, the way you read this. So here's the region, 49 kb region, so we're zoomed out quite a bit further than we were on the previous slide. Here's KRAS down here on the bottom, so exon 1 down at this end, so this is on the minor strand, going from right to left. Whole genome sequencing, you just get a sea of reads, so all those gray sequencing reads I showed you earlier, each of those just shows up as just a little tick on this mark, on this display, because it's zoomed so far out. Exome sequencing, we've essentially used a reagent that will allow us to only focus our sequencing reads on the exons of interest. This is where the coding mutations live. This really is much more cost-effective. You don't have to look at the reads and the intervening regions that you may not be interested in if your question is specifically around coding sequences. And RNA sequencing, you let the cell do the exonic selection for you. So in this case, the cell has spliced out the introns, so all of your reads are only mapped to exons as they should be. You also get the added information of what exons are attached to which exons, and this is also quantitative. So if you have very, very high coverage of specific genes, those genes are expressed at a higher level, and genes that have low coverage are expressed at a low level. I use the phrase whole exome and whole genome sequencing. That's really a bit of a misnomer. They really don't actually measure anything, or probably more specifically, we don't have great bioinformatic methods to assign absolutely every single sequencing read uniquely to a region of the human genome. This is actually a slide from my own work working neuroblastoma. In this case, each little tiny slice here is a neuroblastoma tumor. And the y-axis here is a single exon. So each little slice going this way is an exon. One means that we are completely confident in where those reads were aligned and our ability to find mutations in those exons. And so these are sort of the regions where we had no doubt we can find mutations, we have good coverage, we're well powered. As you get towards the bottom of this plot, you can see our confidence on mutations essentially drops to 0. Not just in the exome sequences, which are in these purple, but also in the whole genome sequences as well. This is really sort of a blind spot in the genome where they're highly repetitive, our reads are short, we just cannot be confident that in our ability to find mutations necessarily in this region. The other interesting technical aspect is that the blind spots in the genome are often, down here they're essentially the same as exome sequencing, but often they're actually not exactly the same as exome sequencing due to differences in read length, mapability, and just sheer depth of sequencing. With exome sequencing, since you are able to focus the capacity of your DNA sequencer on a relatively smaller genomic footprint, you can now sequence those regions to much deeper coverage and might have much greater confidence in the mutation calls at each of those bases. So I still use the terms whole genome and whole exome sequencing because the concept is you're really trying to be as comprehensive as possible, but it does not necessarily mean 100% of the genome or exome analysis. So at the time, it was about 80% of the whole exome was covered at, in this case, was only 14 exomes needed to be called covered. I think that number has probably gone up to 90 plus now. But I think there's still a lot of room for a lot of sort of custom panels to look, especially at these hard-to-read regions, and to complement not just Illumina sequencing with other sequencing technologies, longer read sequencing technologies that basically generate much larger reads that are much easier to map across these hard to capture in sequence regions. I have one slide on PacBio. I'm not going to cover those technologies in great depth actually in this talk. Yeah, rarely. I don't really know any clinical labs who are currently using those technologies due to problems specifically with background error rate. In clinical labs especially, we're quite focused on point mutation detection. Those technologies have very high background point mutation error rate. I think they are going to find a place at some point of resolving large structural variation, but they really need to compete on not just sequence quality, but also cost as well. Certainly dollar per base is very hard today to beat those. So that's the data that's by far the most ubiquitous. I wanted to just give some context to what RNA sequencing data looks like. This is actually a plot I like to make for all of our RNA sequencing data sets. So for a single patient, I like to just plot all of the exons and just say what is the expression level for all those exons, just to sort of get a feel for what's the dynamic range of our data. I actually do this for almost every data set I get. Before I really do anything, I make a distribution. I look at what is the shape of the data. And I show it in this case because this is actually data from a breast cancer patient, and we want to know what type of breast cancer do they, are they ER positive, are they PR positive, what specific markers are they expressing and at what level. And what we want to do is take the single patient, really just compare it to two reference sets, a tumor from an ER positive breast cancer and ER negative breast cancer. The colors are really not showing up great here. In this case, here's the ER, right here in red, here's the ER transcript, and you can see the corresponding transcripts down here in the ER negative breast cancer are really quite low. They're really well below the threshold here. And the patient as well, basically what we want to do is look at, and you can see it. The story here, we essentially want to take these reference genes, look at their level in the patient, and basically compare those to reference sets as well. So if these dots are higher up in the patient and correspond to a specific threshold in a training set, they are positive breast cancer, ER negative. The third type of cancer genome variation is probably being used more and more, being read out more and more by next-gen sequencing is epigenetic modification. In this case, this is another IGV screenshot. I've shown MLH1 here along the bottom. Here's the promoter that controls this gene, and in this region, methylation of this promoter very strong control over expression of that gene. So in this case here is normal tissue. This promoter is unmethylated, and this actually results in normal expression of MLH1 in normal tissue. And here's the exact same type of data, the exact same assay run on endometrial carcinoma, and you can see the promoter has essentially turned completely red. So what's been done in this experiment is they've taken the DNA, they've treated it with bisulfite, and that converts all of the unprotected C's into a T. So this is into a T, and this is really indicative of essentially a promoter with a whole series of C's in the CpG island are unmethylated, and therefore with result in normal expression, normal tissue, but silencing this gene in cancer. Just like mutation calling, this assay is completely quantitative as well. So you can see this promoter is methylated in this tumor, but you can actually see a sort of a shadow of blue signal as well. And this is still persisting from normal cells that are also present in the same tumor tissue. Sort of a cheat sheet great for sort of exams, sort of coming up with the experimental design. Really, none of these assays really stand completely alone. Genome sequencing is great for sequences, copy number alterations, etc. I'll have to go through this in great detail. But really think about how you not just read out these sources of cancer genome variation in one shot, but really start to think about integration of these types of mutations. So the experimental design I'll use quite frequently is actually a combination of both exome and RNA sequencing. Exome to get the sort of the parts list, what are the mutations, what are the copy number alterations. I'm going to talk a little bit more about purity and ploidy, how do we have sort of challenges in working with clinical samples. But not only what are the mutations, but what ones are actually expressed, which ones are actually being used by the cell, which ones result in loss of function or loss of expression of a certain gene, which mutations are expressed at a very, very high level. Methylcene as well being very useful for looking at regions of genome variations that can result in loss of gene expression, but you're completely blind to using exome sequencing. So I'm going to go through this list sort of one by one, so the next sort of series of the talk is really just